Hello, everyone. I'll introduce the paper submitted to iMeta. The title is Gut Microbiota-Mediated Immune and Metabolic Dysregulation in Coronary Artery Disease Progression and Prognosis. Coronary artery disease remains a leading contributor to the global burden of disease. It is a dynamic pathological process in which clinically stable coronary plaque may progress to acute myocardial infarction through occlusive thrombosis. Accumulating evidence implicates the gut microbiome as a non-traditional contributor to CAD. Microbial influences are thought to be mediated through metabolic pathways and bacteria-associated molecular patterns. Building on our prospective cohort study, we integrated metagenomic, metabolomic, and clinical approaches to evaluate the prognostic relevance of coronary artery disease-associated microbial features. We further investigated whether disease-associated microbiota may contribute causally to atherosclerosis progression using fecal microbiota plantation. Together, these analyses aim to define a microbiome-based signature of coronary artery disease severity and prognosis, and to establish a translational framework for incorporating metagenomic profiling into cardiovascular risk stratification. 59 control individuals and 260 patients were enrolled. Detailed clinical information were collected. Patients were stratified according to the number of stenosed vessels, atherosclerotic burden, myocardial infarction ratio, and cardiac troponin levels increased progressively. Shotgun metagenomic sequencing were performed, and generated 1,046 MAG. The global structure of the gut microbiota differed markedly across CAD stages. Since bacteria often function as guilds, we clustered these MAG into 36 CAG. CAGs enriched in CAD were mainly composed of bacteroids and were positively associated with disease indicator. Since circulating metabolites act as intermediaries between the gut microbiota and cardiovascular biology, we performed multiomics and cross-domain association integrating disease phenotypes and microbial functional modules. Microbial LPS biosynthesis strongly correlated to CAD progression, and aromatic acid metabolism associated with thrombotic event. Next, we follow up the cohort. 43 major adverse cardiac events occurred. We constructed random survival forest models using microbial features to determine the prognostic value. We noticed CAG35 comprised of bacteroids thetaioteomicron and aromatic amino acids pathway were prioritized drivers. Moreover, baseline serum levels of phenylacetic acid and indole 3-carboxylic acid were also predictive of MACE event. Overall, microbial predictors provided additional prognostic information combined with conventional risk factors. To investigate whether CAD-associated microbiota can contribute causally to atherosclerosis, we perform fecal microbiota transplantation experiment. Notably, aortic histopathology exhibited severe atherosclerotic lesion in disease mice. In parallel, fecal LPS levels and protherogenic signs were elevated, and vascular expression of toll-like receptor 4 and coreceptor CD14 was also upregulated. In conclusion, CAD-associated microbiota can transmit atherosclerosis susceptibility. Since gut microbiota induce systemic and vascular inflammation, which persists throughout the entire pathophysiological process of atherosclerosis. We found increased proportion of CD4 T cells and neutrophils in the aorta of disease mice, with exhibited expansion of immune activation characterized by elevated Th cells and reduced Th2 cells. In this prospective cohort with follow-up, we integrated multiomics profiling to dissect how microbial variation relates to CAD progression. Gut microbiota-derived aromatic amino acid metabolism was associated with thrombotic risk and MACE events. Through animal experiments, we demonstrated microbiota contribute to atherosclerosis. These data support the development of microbiome-based strategies for future research.